Gametogenesis. Today we will see the gametogenesis, and we will only see sperm formation. The primary sex organs in the male, that is testis. Testis is the primary sex organ in the male, and the ovaries are the primary sex organ in the females. And testis produce sperms and the ovaries produce ovum respectively. By the process of gametogenesis. In testis, the immature male germ cells, that is spermatogonia. Look at this, spermatogonia is the immature male germ cell, and it produces sperms by the spermatogenesis that begins at the puberty. The spermatogonia, which is in singular form we call this as spermatogonium, present on the inside wall of the seminiferous tubules, multiply by the mitotic division and increase in numbers, and each spermatogonium is diploid and contains 46 chromosomes. Okay, spermatogonia present inside the wall of seminiferous tubules, and it multiply by the mitotic division and increase in numbers, and each spermatogonia is diploid and contains 46 chromosomes. Some of the spermatogonia, called the primary spermatocyte, periodically undergo meiosis. There are too many spermatogonia. From there some differentiates and undergo meiosis and form primary undergo differentiation and form and we call that spermatogonia as a primary spermatocyte, and they periodically undergo meiosis. A primary spermatocyte complete the first meiotic division, that is reductional division, leading to the formation of two equal haploid cells, that is secondary spermatocyte. Okay, there is some notable point. Okay, that is that primary spermatocyte which is diploid, and the secondary spermatocyte is haploid. Okay, we have only 23 chromosomes. As we know that secondary spermatocyte is haploid, then it is obviously that it contains 23 chromosomes, and the secondary spermatocyte undergoes second meiotic division, and the secondary spermatocyte then undergoes second meiotic division to produce 4 equal haploid spermatids, and the second meiotic division is equational division. There is no any reduction in the ploidy, and that's why spermatids is also a haploid, and we get 4 haploid spermatids. And what would be the number of chromosome in spermatid? As we know that haploid, that is 23. The spermatids are transformed into spermatozoa by the process called spermiogenesis. This is so important and confusing. The spermatids are transformed into spermatozoa by the process called spermiogenesis. Okay, after spermiogenesis, sperm heads become embedded in the Sertoli cells. When the spermiogenesis completed, the sperm heads are embedded in the Sertoli cells, like this here. This is Sertoli cells, this is Sertoli cells, this is Sertoli cells, and the spermatid which is attached to the head of the sperm head is attached to the Sertoli cells. And finally, when the sperm released from the seminiferous tubules by the process called spermiation, when the spermatid release from the seminiferous tubules, or we can call it the Sertoli cells, then this process is called spermiation, and which is also important, spermiation. Okay, Spermatogenesis start at the age of puberty. As we know that in the male, spermatogenesis start at the puberty, puberty, which means the age when the humans become reproductively active, and in the male it is, I think, 12 to 13 to 14. Okay, or spermatogenesis start at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone, which is of hypothalamic hormone, and they increase the level of GnRH, and it is the hypothalamic hormone, and if when the levels of GnRH increase, it acts on the anterior pituitary and stimulate the secretion of two gonadotropins, that is luteinizing hormone and the follicular stimulating hormone. And the luteinizing hormone acts on the Leydig cells and stimulate the synthesis of androgens. Androgens in turn stimulate the process of spermatogenesis. Androgens spermatogenesis को स्टिमुलेट करता है. FSH acts on the Sertoli and stimulates secretion of some factor which help in the process of spermiogenesis. FSH सर्टोली सेल पे एक्ट करता है और स्टिमुलेट करता है सिक्रीशन ऑफ सम फैक्टर्स which help in the process of spermiogenesis. Let us examine the structure of a sperm. It is a microscopic structure. Okay, now we will see the structure of a sperm, and it is a microscopic structure which we cannot see the sperm with naked eye. Okay, look at this. This is head region, and this is neck region, and this is middle piece, and this is tail region. हेलो, हाँ मिस, चलिए ना जाइएगा आज 5 बजे. हाँ चलिएगा 5:30 में, क्या दिक्कत है? ठीक है. A plasma membrane envelops the whole body sperm. The sperm head contains an elongated haploid nucleus, and the anterior portion of which is covered by a cap-like structure, acrosome. The acrosome is filled with the enzyme that help the fertilization of the ovum, and the middle piece processes numerous mitochondria, which produces energy for the movement of tail that facilitate the sperm motility essential for fertilization. The human ejaculates about 200 से 300 million sperm during a coitus, of which for a normal fertility, and at least 60% of the sperm must have normal shape and size, and least 40% of them must show vigorous motility. Look at this. This is the structure of a sperm. Here is neck, and here is middle piece, and here is a lot of mitochondria which provide the energy for the movement of the sperm, vigorous movement of the sperm. There is a plasma membrane which is covered the whole body of the sperm, and the acrosome which contains some enzymes which helps in the during fertilization, and nucleus contain chromosome material. Okay, and for the normal fertility, it must that the 60% of the sperm shows vigorous motility. That is 60% of the sperms has normal shape and the size, and at least 40% of the sperm have the vigorous motility. Okay, sperms are released from the seminiferous tubules. Sperm seminiferous tubules से release होता है and are transported by the accessory ducts. There are a lot of accessory ducts we will discussed earlier, like the secretion of epididymis, vas deferens, and seminal vesicle, and the prostate are essential for the maturation and the motility of the sperm. Look at this. I hadn't observed that secretion of epididymis and vas deferens. I thought that epididymis and the vas deferens is just a duct which has only conduct, but there is written that epididymis also secretes some things and the vas deferens also secrete. Okay, the seminal plasma along with the sperm constitute the semen. सेमिनल प्लाज्मा के साथ साथ स्पर्म को हम लोग मतलब कि दोनों को मिला के हम लोग सेमेन कहेंगे। द फंक्शन ऑफ द मेल एसेसरी डक्ट एंड द ग्लैंड्स आर मेंटेन्ड बाय द टेस्टिकुलर हार्मोन्स। फंक्शन ऑफ मेल एसेसरी डक्ट और ग्लैंड्स जो है सो वो मेंटेन होता है टेस्टिकुलर हार्मोन्स एंड्रोजन से। द फंक्शन ऑफ द मेल एसेसरी डक्ट्स एंड द ग्लैंड्स जो कि मेंटेन होता है टेस्टिकुलर हार्मोन्स एंड्रोजन से। ओके, द प्रोसेस ऑफ फॉर्मेशन ऑफ मैच्योर फीमेल गैमेट इज कॉल्ड oogenesis, उसको हम लोग oogenesis कहते हैं, विच इज मार्क्ड। ओके, oogenesis we will discuss later.